Well, thank you very much for having me. I think these kind of conferences dedicated to specific monogenetic disorders are a real revolution in the field, especially because we join with parents and patients here. I've even been in one lately where industry was also present and the EMA that was even more integrated. But I think this is a very good step, and that exchange is very crucial. Then again, talking to both parents, molecular biologists, clinicians, is a difficult task to make a very appropriate presentation. So I hope I did okay. I even modified it today whilst listening to you and think what would be a good contribution to everything added already or said already. And I think one of the things that we really try to do, Kim is actually my clinical boss, so to speak, and we're together in the NSU team. And every week we have what we call a multidisciplinary meeting from the care perspective, not so much a research perspective, in which we really try to dissect every level of rare diseases or individual patients. And I think one of the most important learnings is that even if you have cohorts defined by GRIN2B or STX-PP1, the ones that we study, that we struggle so much to understand variability and heterogeneity, not only in terms of severity, but some people already mentioned, especially in time. And I like always this sort of this representation of how everything connects to everything, and that nothing is unidirectional, and something that happens in one level affects the other, and that the prenatal stages and the postnatal stages, how they interact, especially with effects of the mutations at different stages, is very important to understand. So I think to start at our mission at this NSU Neurodevelopmental Precision Center, as we baptized it 3 or 4 years ago at the Amsterdam UMC and the Emma Children's Hospital, is really to try to acknowledge individuality in every essential sense of the word. And it's a very nice hospital. The building is old, but the Children's Hospital has been rebuilt a couple of years ago, and we often have, it's a very cheerful and lively hospital. This is Ajax visiting. I'm not an Ajax supporter, by the way. I'm Feyenoord. But it's a very, I think it's a very stimulating environment where we really get the freedom to develop these new thoughts. And if I would summarize all of that, then I think what we really try to do is to advance towards the best possible treatment for the individual child. So every time think, what at this moment with knowledge present can we implement, and are we certain of against what has already been done for ages and not working good enough. And we identify sort of 3 components in that strategy: is to inform, so to integrate information around the individual, data and information, not necessarily the same thing, to inform or to create decision support to understand what should we do. Sometimes it's just the genetic mutation, but sometimes it can be the combination of many other elements of information, and to perform the treatment. And there, I think, is another very exciting thing related to this conference, is that we are moving away from traditional trial designs to more what we call the N of 1 designs, in which every child is his or her own control. We have not sufficient numbers to randomize, and we don't want to randomize because we're not looking for averages. We're looking for the individual effect. And the third component is to develop, which has always already been discussed with skills and also by Caitlin, is biomarkers and patient outcomes, so clinical endpoints. How do we actually see that the treatment that we think should have effect on a certain mechanism had that effect, and did it really result, or was that effect actually related to improvement, or can we actually detect improvement? And we are not a GRIN2B center, so to speak. We need to serve more children. And I always like this picture. This was from a Cell paper in 2020, which sort of accumulated the evidence from 10,000 whole exome sequencing analysis. So the investigation in which every coding sequence is evaluated for mutations that are not present in normal cohorts, so to speak. which resulted in the combination of about 102 genes. And what they did in this paper was, I think, quite instructive. First of all, they wondered what is the timing of these 100 mutations? Are some more early, some more late? And then they came with this sort of distinction of the purple and the green ones, that the ones that are relatively more early in effect are not genes that have a distinct effect like GRIN2B, but they are more transcription factors or mutations that affect a multiple number of other genes, and therefore are also, if you look at this representation, more associated with more severe, or let's say early neurodevelopmental disorders, not really restricted to autistic phenotypes. And the other thing is that what they designated in this paper by studying the functional and the phenotypic distinctions known from bioinformatic information on these genes that were identified by the sequencing, is that the genes like GRIN2B that have distinct effects that they call neurocommunication genes are relative, or also both active before, but also have an effect after birth. And if we think about treatments, especially these curative treatments, it's very important to understand or determine whether we will be treating the pre or postnatal effects, and what happens if we can only treat the postnatal and not the prenatal effects. But the most important thing, and I'll be bothering you for the majority of this talk, is the right part here, is that if you would seek convergence or common pathways across these monogenetic disorders, or these genes that have sufficient impact to call them actually monogenetic forms, so to speak, is that they all have some kind of effect on what they would call excitatory inhibitory cell lineages or cell functions. And from that, and that resonated with this long-standing theory that the EI balance, so the balance between excitatory inhibitory signaling or excitatory inhibitory cell functions is a convergent pathway, and the disruption between E and I might be something of a common theme to study and to understand the convergence, but also the divergence between monogenetic disorders. And that last part is something that we have taken as sort of a handle or a framework to study several of the genetic disorders that we are focusing on, like GRIN2B, but also STXBP1. That's a very different type of genetic disorder, has to do with physical docking of neurotransmitters and not with like NMDA receptor components. But to see whether we could use that framework to gain understanding. And why that is important, I don't know whether all of you, I'll explain a little bit more about the EI balance, but before I do that, I think one of the most striking or revealing things to me was when I read this book of George Buzsaki. He's a very famous Hungarian neuroscientist, that the end stage of development can be seen as how spontaneous brain activity is actually constructed. So 95% of our brain activity is sort of spontaneous and not contributing to function, which is very counterintuitive. Has been shown in many species. And his theory, and that's why he calls it the brain from inside out, is that bulk of spontaneous activity is actually the organ or the mechanism that supports or governs brain functioning. And if things go wrong in development through genetic disorder or premature birth or toxicity or combination thereof, one might think that construction and that function of spontaneous brain activity is actually perturbed. And why is that important? Because the theory is that spontaneous brain activity organ, or that bulk of activity to support brain functioning, can only do so when it has that balance between excitation and inhibition. That's one of the most instrumental ways for plasticity and successful resilience of the brain to respond and regulate organs. And I think that way of thinking creates a different outlook on the relationship, for instance, between pre- and postnatal effects of genetic mutations, for instance. And in a more functional way, because not every one of you will be familiar with this phenomenon of excitation inhibition, you can actually see it also from inside out. So this is a picture of neural activity, spontaneous neural activity, may seem chaotic and transgressing through axons and then dendrites through the cell body. But that every time the decision whether a cell permits the signal to go on is a very complicated decision, and it is actually the accumulation of many synapses and many parts of information as it gets from different cells and different circuits nearby or more distant. And the machine chamber, so to speak, of those decisions and the way the brain can actually compute or sustain that balance is the synapse or the synaptic cleft. And if you zoom into that, then you see all these proteins and all these systems and pathways that are contributing and modulating that. So there's an extremely complicated situation that at the end of the day cannot result in something very simplistic like excitation or inhibition. And one of these proteins, obviously, or actually components of the protein complex is GRIN2B, for instance, or GRIN, or other GRIN components that then dysfunction. And what you see here, that's a bit of an old picture, but I really like it because all the little stars, I don't know whether you can see them here, are examples, like here, if you see the MDMDR, of existing treatments, so repurposing candidates that we could use to mitigate the effects of mutations that affect some of these proteins. And then the red synapse is the excitatory synapse, and the blue one is the inhibitory one. But the funny thing is we did not really get around to testing them because we didn't know which child has which protein deficit and which agent should we then test. But now we have the situation that we're more focusing on these specific cohorts of which we know what is the protein at stake and that we can really do very targeted trials, but we don't know exactly how the surrounding machinery will respond and whether all kinds of compensatory or other types of homeostatic interactions will occur. And that's what I started, that even within children with the same mutation, the genetic background and context will determine what will happen down the line in the machine chamber to actually govern the effects of pre and postnatal development. To go back in simple terms, the reason why we originally inferred that hypothesis of excitation inhibition obviously is that we know that like GRIN2B is associated with a lot of sensory sensitivities, epilepsy, seizures, epileptiform activities, which already indicates that something is wrong in the maintenance of brain activity. So how can we use that to our advantage? Now, I think, and that was therefore I was really inspired and intrigued by the talk about the blood and the homeostasis of iron and the remarks we got about glucose, that if something is perturbed in this system of maintaining, for instance, a neural balance, it cannot go without an effect on other systems and vice versa. I think that's something we really need to keep in mind, that all these homeostatic interactions will take place. And in the Bujaki phrase of the word, the EI balance really tries to create a stable situation at a given physiological set point. And if that physiological set point is altered by GRIN2B, all kinds of other systems will try to create or go back to the physiological set point, which in turn will have a vicious cycle and a sort of feed-forward inhibition status. So I think this type of picture, it's actually from an Alzheimer paper, but I think we end like we begin, so it doesn't really matter. Developmental disorders or neurodegeneration, they have the same type of mechanisms. It's important to think about the interaction between the neural homeostatic and the more physiological homeostatic type of interactions. And alongside that thinking, we have created, or we were thinking, how can we use this? Because we cannot access brain tissue. We cannot immediately study how that has resulted, all these homeostatic interactions in the child brain. But we can use theories that describe how, as Bujaki called it, that balance or the spontaneous activity has organized itself. And one of those theories, I won't bother you with the complex statistics of that because that will take a couple of hours, is called criticality. You can look it up, and criticality is a mathematical theory that either earthquakes, mass extinction, birth locks, once a system is very complex, it tends to organize itself in a certain equilibrium or balance in which it is between order and disorder. And that balance has a certain mathematical given or a mathematical definition that one can use and apply, for instance, also to brain activity, to ask yourself, does the brain or the spontaneous brain activity that we then try to measure with resting state for as far as we can get to resting state, but let's assume we can, that balance also, that the brain also tends to organize itself into that balance or that critical state between order and disorder in which it's maximally resilient to perturbations from the outside. And if something happens in development like a GRIN2B mutation, one would expect that that balance is offset. And the interesting thing is that it's quite a directly applicable mathematical theory. And what I try to show here in this diagram is that this inhibition-dominated signal, so this is a continuous EEG signal, which is a sort of has not much variation, both in terms in time and in power. And this, the middle one, is very variable, both in terms of power and time, and the bottom one is sort of white, nearly white noise. There's too much power and also less variation. That one can use it to extract a parameter of whether that system is balanced, yeah? And we've done that, and it's an algorithm. So this, if it's inhibition dominated and you calculate that mathematical function related to that brain theory of criticality, that the number below 1 indicates that the signal is relatively inhibition dominated. If it's around 1, it's balanced between excitation and inhibition, and if it's above 1, it's excitation dominated. And from that, we've created a biomarker, which we actually yesterday got patented after 9 years. So that's what it takes. Yeah, that we were quite happy with that. And not for our own money, by the way, but just to get the money from the right guys. That's only a reason why we do it. But we apply this, for instance, in this simple initial between autism and TDC, not to show that autism is related to hyperexcitability, but to make the opposite point, that some children with autism have pervasively inhibition-dominated brains. Some children have overexcitable brains, and some children, if you average it of all electrodes, have balanced brains. So the heterogeneity in physiology is very extreme and very remarkable. And we cannot just give Ritalin or antipsychotic to any child without taking these kind of elements of homeostatic brain regulation into account. And yeah, I cannot go into all the, how much time have I now? Where's the stopwatch? Oh, yeah. Minutes into all the details, but to summarize, we've applied that to a number of situations, also to a number of genetic disorders, tuberous sclerosis, GRIN2B, STX, Kleefstra. And what was very interesting, that if you would think all these children with their susceptibility for seizures and epilepsy, that they would tend to have a hyperexcitable or excitation-dominated brains. The opposite is true. They have very slow inhibition-dominated brains, which are, and I think at the end, the more we think about it, it's quite logical. There's no room for information processing, so it's very limited in terms of variability. And whether that's due to a lack of energy resources, whether it's due to incomplete maturation of or specificity of circuitry, we don't know. But it's the beginning of something very important, that there is an overlap, and that this inhibition-dominated brains, these slow brains, as I showed on the top diagram, is something that is shared by children with profound mental retardation. And children with a little bit less mental retardation and more dyslexia, different type of neurodevelopmental disorders, not the severe genetic ones, tend to have the excitation-dominated brains. And I think that's an important beginning of stratifying very different. That's why I asked about the autism, the Simons Foundation. Are you focusing on autism or neurodevelopment? Because I think they're very different things. And if you want to read up on it, we have proven in several, these are two examples, that we use it either to stratify, for instance, children with a specific genetic disorder, but we've also tested it in certain trials. We have a very big interesting bumetanide. I cannot go into that today. It's a repurposing agent in which you can correct, or hopefully correct, GABAergic inhibition by chloride, the influence in chloride regulation. And another thing is that now that we have this EEG, it's a good translational instrument in consortia in which we go from stem cell level of EI towards clinical patient level of EI and compound screening, so to speak. So we use it as the link between all these translational levels and to understand how genetic effects cascade into circuit or network level defects or behavioral cognitive changes. And just to quickly show you how we do that, we have some new fancy cognitive network type of assessments. Kim will tell you more about how she looks at the whole relationship and the implementation of cognition. We have PROMs, so measures of sleep and energy that we can administer very quickly, weekly, for patients to really sensitively monitor the changes in effects of treatments or states anyway. And what we really find important, that once we have all that information and we think about even one framework, that we can together discuss this in dashboarding. So we have preconditions about pregnancy, mutations, but we have also the reasons why development could be altered towards measures of EEG cognition that we can follow. And we have a diagnostic dashboard for that, and we have also a monitoring dashboard that we can also together visualize what has been happening. Is it more an EEG responder, more a cognitive responder, or both? The prompts are improving, but why is the other one lacking behind? Well, you have two similar children, etc., etc. And I think that's very important to generate hypotheses and to really go into understanding what happens when we start these new kind of treatments in these new kinds of cohorts. And another thing that is very important is that we train our students that if they go into new territories of patient populations that are defined by genetic disorders, such as GRIN2B, that we say you need to create a mind map that of the genetics, molecular, and functional and clinical findings, and that they have to train themselves to make this kind of pictures. This is GRIN2B, for instance, in which they get a flavor of the gain of a loss of function, and molecular, and everything we've been discussing today. But they really train themselves to go into the literature and to know what is known. And this mind map for GRIN2B is, for instance, completely different than the one for GRIN1 already. And they need, I think, in terms of teaching, we need to start training them in that capacity, that they know what's behind the mutation when they study all these kind of aspects, the genetic aspect, the molecular aspect, and the functional aspect, and the clinical aspect. Now, if then they create summaries that GRIN2B is well characterized, a monomolecular deficit with with loss of and gain of function differential manifestations, and that they have to think what's the best possible treatment. And then we create the hypotheses in our MDO about what we actually think, what do we expect to see when we will apply, for instance, EEG, and what will be the effect, for instance, of this mutation. That's where I asked about the inhibitory and excitatory effects earlier on, to see what we will find if we apply the whole network or the brain level type of measurements. And indeed, this inhibition dominated the eye, that one could infer from what is known about the molecular or the more genetic and functional aspects of GRIN2B in literature, that we then test that. And I just got this information yesterday, so it's not perfect, but I just wanted to show how that pans out. So we had 5 patients that were age-matched with TDCs, and we performed the EEG and to visualize source-reconstructed biomarkers. And initially just some interesting features about some having macrocephaly, some being mild, some being severely affected, some having different types of mutation, but we all focus on loss of function here. And then you get this kind of pictures. Unfortunately, they were not calibrated yet, so we cannot very easily look at the differences. And this is from top to bottom, absolute power, relative power. This is a measure of entropy, and this is the EI measure that I showed you. Here are the controls, and the red is eyes closed and the blue is eyes open. And most children, unfortunately, with GRIN2B are not capable of doing eyes closed. If we can get EEG anyway, which is a task on our own, we have very well trained people that can comfort a child, and we have the wet cap, and it goes. And we also, by the way, like Caitlin, we also have a bus, so we can also go towards the patients. That is also indeed a very good solution in many times. But just to show you if we take the alpha frequency band for these results, then and it's a bit difficult to show you all the details, but just accept from me that in 4 out of 5 patients, again, we found this prevailing inhibition dominated brains across most frequencies, which is accompanied by excessive delta activity. And a delta activity is actually normally only present in sleep or in coma or in severe brain damage. And that has always been neglected actually in neurodevelopmental disorders because it was always case control studies. But I think it is trying to tell us a message. Either it's persisting immature brain activity, because in the womb, there's mostly delta activity also, or the children are sort of sleeping whilst awake. And we need to understand what that is, because related to these energy and these metabolic problems that we discussed, I think there could be a connection there for which we can use that to be quantified by EEG. And then to finish, and that's maybe my most important, it's not so much an opinion, but it's a question that once we go into these dedicated meetings, that we have the cure, the care, the disease modifying, the disease altering aspect, and that we need to be aware how much is feasible and how much resources, money is spent to more the left side, to really the gene therapy, the small molecule therapies, or the more repurposing to support what we can do now. So that discussion and that balance is, I think, very important to be connected to what type of research do we have, what type of knowledge do we have, or what is best possible at this moment, 5 years, 10 years from now, and are we honest about, for instance, rescuing gene expression, how feasible that is in a fully developed brain, for instance. And I think we need to start in these kind of conferences, debates about that, because that will be very instructive, and also things about parent support and all kinds of other aspects that might deserve science or support anyway. It needs to be taken into account. So, and we are at this moment completing our own L-serine multiple N-of-1 trial, because that's our philosophy, as I said. So this is a genuine ABA design. And I think we will have the first data in, I think, 6 months. So hopefully in the next GRIN conference, Bibi's and Hollander, she's the PhD, she can present the results. The EEG results I just showed you are also of that children. And so just to give you an overview, this N-of-1 trial business, we do that also for like CBD oil with Fragile X, and L-serine also for STX-BP1, because the interesting thing is that positive allosteric modulation indication does not only exist for GRIN2B4, but for many other genetic disorders too. So that could be interesting. And in more generic cohorts from diagnosed by autism or sensory sensitivity or ADHD, we use single case experimental designs, which I think is also a very attractive design to avoid the old school RCTs, and to really treat patients on an individual basis, and to take into account and to really follow in participation with patient and parents the effects or the developments of effects. And the last thing that I think is very important, I heard the term natural history coming. I think that's very crucial, that we have one natural history study for each genetic disorder with as many people on board and not 6 different ones. And that we need ICT or digital techniques. I'm working with Accenture to really think, how can we create data interoperability because we have so much different types of information of a relatively small number of patients. How can we connect it both semantically and technically? And that we can really, at any time that we want, flexibly browse our own cohorts. How many children with middle ear infection did get anti-epileptic effects and were with GRIN2B or GRIN1, etc., etc. We need that type of flexible systems to dashboard and to browse our cohorts, and to come to the right type of intervention and hypotheses. So that was actually what I wanted to show you about the NSU. So we advocate clinical data integration or interoperability, N of 1 trials, and to create these decision support systems. That would basically summarize what I was trying to say. There's a lot of people involved. I cannot name all of them, but I think team science obviously is the most important instrument for these kind of rare disease activities. So I would like to thank all these people and also you, because I think it's a great thing that we're here together. And if you have questions, I'll be here. Thank you.